Hello student, today I'm going to explain about the process meiosis, and I think you know about the process of mitosis. I have started from the topic meiosis because it is actually complicated process than mitosis. We know what meiosis is, a reductional division. What I told, a reductional division. If simple question will come in exam, define meiosis. So you have to write appropriate definitions. What will be the definitions? And the process meiosis actually started from the diploid cell. It really started from the what? Diploid cells. That diploid cell is also called as what? Meiocytes. That diploid cells are also called as what? Meiocytes. Meiocytes singular. Now, that meiocyte cell will undergo two successive divisions. What I told? Two successive division. One will be meiosis 1 and another will be meiosis 2. So, through meiosis 1, that cell converted into what? N and N. Clear? That means what? Here you have seen the number of chromosome has declined from 2n to n, and number of cell also increased, isn't it? And this, from this n number of cell, while meiosis 2 will start, then again you will see two number of cells will form from each daughter cell, which will be formed by meiosis 1. Clear? That means what? You have noticed or not? Let me label it. This is meiosis 1 and this is meiosis 2. This meiosis 1 is actually, you can call it, it is as a reductional division. Reductional division. And this meiosis 2, you have noticed or not, the number of chromosomes remain as it is from here to here. So this meiosis 2 is called as what? Equational division. Equational division. So that means what? You have seen that in this process, two successive division has taken place, started from diploid cells and converted into haploid cells. And to enter into the actual process of the meiosis, how the chromosomes converted into 2n to n and n to n again, we need to know about the ploidy. What I told, we need to know about the ploidy. Ploidy means what? You can write the number of complete set of chromosome. What I told, the number of complete set of chromosome is called as what? Ploidy. The number of complete set of chromosome is called as ploidy. Now, if it is n, it is called as haploid. Very simple. If it is 2n, it is called as diploid. To know about the concept of amount of DNA, you need to know about the ploidy, and you need to know about the another term which is called as C value. What I told, which is called as C value. Because in NCRT, in this chapter, I have seen the last question, you have to track the ploidy and C value in each phases of the entire cell divisions, or you can say entire cell cycles. In G1, what will be the conditions of ploidy natures of the cell? In G2, what will be the conditions of the chromosomes numbers and C value as well? So you have to know entire things so that only one complete concept will you can gather. So C value is what? C value is the amount of DNA. The amount of DNA in haploid set of chromosome. In haploid set of chromosome. So that means what? In N, the C value will be what? Obviously, N is equal to C. If you compare it, suppose 1N is equal to 1C. If it will convert it into 2N, then it will convert into 2C. If it will convert into 4N, then it is converted into 4C. That is why I am going to tell you the number of complete set of chromosome is called as ploidy. And if it is haploid, it's supposed to be 1N, or 1N not necessary to remember to mention. For understanding purpose, I have used 1N. You can use N. And C value is what? Amount of DNA in haploid set of chromosome. As it is haploid is N, so obviously it's supposed to be N is equal to C. Clear? Now, next we can go to the next step. To know about the meiosis, what you need to know? You need to know about the homologous chromosomes. Let me write. You need to know about the homologous chromosome. You need to know about the chromatid. Okay, what I am going to use, the terms what I am going to use, these things first you need to clear, so that only it will take less amount of time to understand the concept of meiosis. Okay, so what you need to know? You need to know about the homologous chromosome. You need to know the chromatid. You need to know about the centromere. Okay, you need to know about the diad, then tetrad. You need to know about the bivalent. Then you need to know about the crossing over. You need to know what about the crossing over. You need to know about the recombination nodule. You need to know about the synapses. It is not the synapse, which is the junction between the two neurons. It is about the synapses. You need to know about the synaptonymal complex. Okay, so these terms, and while or when you are going to use these things, you have to know. If you have understood perfectly, then the concept of meiosis you will know in better way. What I told you will know in better way. Now let me draw the homologous chromosome. Let me show you the position of the centromere, then position of the chromatid. What are the things? Let me show you. What are homologous chromosomes? That homologous chromosomes are present in a pair which are same in size, okay, same in size, same in shape, same in the position of the centromere, and each of the chromosome will be coming from the parents. So that only what will happen, that in two chromosomes will be from each of the parent, that is one is from the mother and one is from the father. So that only homologous pairs will form. So let me draw the homologous chromosome. And we know about the types of the chromosomes on the basis of positions of the centromere. So the chromosomes which I am drawing, this is what? This is metacentric chromosome. In metacentric chromosome, what will be the positions of the centromere? The positions of the centromere, it is in the middle part. So this is called what? This is actually centromere. And here you have seen what I told, chromatid. How many chromatid? There are two chromatid. Chromatid. Same also in this case, here also it is centromere and this one will be the chromatid. And in cell division procedures, will show you one thing which is called as disc shaped around the centromere, which is called as what? Kinetochore. Kinetochore. Now, during the procedures of the meiosis 1, I will mention about what? The synaptonymal complex. What I have used? What I will use? Synaptonymal complex. That synaptonymal complex acting as a what? That synaptonymal complex acting as a glue. Acting as a glue. Or you can use adhesive. What it will do? Glue synaptonymal complex. Complex. This glue fuse the homologous chromosomes, so that only what will the procedures, the process through which homologous chromosome will pair each other, will fuse each other, is the process is called as what? Synapsis. Synapsis. Okay, so till now what we have seen, we have understood about the homologous chromosome, we have understood about the chromatid, we have seen the position of centromere. Now while two chromatid, how many chromatid in one chromosome? Two chromatid. If there is two chromatid, there will be, this is called as what? Dyad. See, dyad plus dyad will form what? Tetrad. So after fusion of the two chromosomes, obviously there will be combinations of entire chromosomes, so that dyad and dyad will convert into the tetrad. So this one also we have understood. Now each chromosome, if you correlate, it is X as a univalent, each chromosome, then it is also one kind of univalent, and this one also one kind of univalent. So univalent and univalent will form what? Bivalent. Now understood about the synapses, we have understood about the synaptonymal complex. After the formation of synaptonymal complex, there will happen, will explain, although I will explain the entire procedures. There will be recombination nodule will appear. That recombination nodule, this will be called as what? Recombination nodule. Recombination nodule. That recombination nodule will give the pinpoint location of the crossing over. That means what? If I extend this chromosome, suppose this is the chromosomes, and here will be this chromatid will be fused. Okay, so that only what will happen? There will be the crossing over. The crossing over means what? One chromatid will cross with another chromatid of the homologous chromosome, so that only recombination will be taken place. So recombine the genetic materials. I think all the terms I have taught. Recombination nodule helping in the pinpoint location where crossing over will be taken place. Let me draw after crossing over what will be the conditions of the chromosomes. This is the chromosome, suppose. Here will be the chromatid. Here is another chromosomes. That crossing over has taken place. And here will be what? Here will be synaptonymal complex. And here supposed to be recombination nodule. So this is the entire story of the meiosis. These things will be happening in the entire procedures, not actually entire procedures. You will see in the Prophase 1. Maximum questions will be coming from the Prophase 1. In Prophase 1, what you are going to see? Leptotin, zygotin, packaging, diplotin, diakinesis. These are the 5 processes. From the 5 processes only question will come. Okay, before going to tell you about the first phase, let me show you the entire process. What are the phases in the meiosis? See, let me write. This is what? Meiosis. It will be having what? Two successive phases. One is, I'm going to write only M1. Okay, M1 it is meiosis 1, and M2 it is meiosis 2. That meiosis 1 again classified into what? Prophase 1, prophase 1, then metaphase 1, anaphase 1, telophase 1. We know what prophase, metaphase, anaphase, telophase. These are the phases of the karyokinesis. After that, cytokinesis 1. Now in this case, that prophase 1 is having some mystery. Till now what I have shown you, that synaptonymal complex, synapses, crossing over, these things will be happening in the prophase 1. So in prophase 1 you will be seeing that another phases. First phases what you are going to see, which is actually leptotin, then zygotin, then pectitin, then diplotin, and then diakinesis. So these are the 5 phases you are going to see in prophase 1. Okay, so you have seen leptotin, zygotin, pectitin, diplotin, and diakinesis. This sequence you have to mention, otherwise some ambiguity may cause in your conceptions. Now in case of meiosis 2, this is very simple. Let me take another place. Meiosis 2. In meiosis 2 it is very simple. You will be going to see prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis 2. See, after cytokinesis 1, the cell will remain at rest for a few minutes of time. So, the resting phase from meiosis 1 to meiosis 2 is called as what? Interkinesis. What I told? Interkinesis. Clear? So, we have seen what are the phases in the meiosis. Now, one by one, I am going to explain. So, let's see that in leptotin. So, let me draw first the cell. In that cell, you have to think whether that cell will be having nuclear membrane or not. Obviously, that cell will be having nuclear membrane. And in prophase, in prophase of mitosis, at the ending of the prophase, what we have seen, that nuclear membrane has disappeared. So these things will be happening in the in the entire prophase. But from the very beginning, what I told, leptotin and zygotin are the shortest phases of the um, Prophase 1. So here let me draw the nuclear membrane. So let me take the chromosomes. Okay, that chromosome will condensed more in this period. Okay, so and the centriole, number of centriole also will be what? 2, because in S phase, in the synthetic phase, the centriole also replicate and that DNA also replicate. Actually, in case of centriole, we cannot use the term replications. It's supposed to be duplications. It's the term replication specifically you can use for the DNA, because in the molecular basis of inheritance, while you will be in class 12, you will see how the semi-conservative model of replication has taken place in case of DNA. Okay, so that centriole start to move in the opposite pole. If it is the pole side, start to move but not gone. And what will happen? Here the chromosome will condensed, means not properly condensed. Condensation has started, or spiralization also occurring. That is why it is that phase is called as spiring phase. Okay, and so that in this phase that chromosomes are visible in light microscope. So these things is happen. Chromosome begin to condense, become visible under the light microscopes. Compactions continue throughout the lactoting phase. Obviously, chromosome compactions. Centrioles start to migrate to the opposite pole. And the terms, phylums, these terms specifically you can use for the lactoting phase or the bucket stage. You have seen these are look like the bucket. They are attached in the nuclear membrane. So this is all about the lactoting or bucket stage. Now next one is zygote. So let me draw another thing which is very very important. I have skipped. In this cell, can anybody guess what will be the ploidy of the cell and ploidy nature, and what will be the amount of the chromosomes or C value? Obviously it will be what? It will be 2n and it will be 4c, isn't it? So the cell which has entered into the meiosis 1, first of all it will be having what? 2n and 4c. Why 4c? Because the DNA amount has doubled in S phase. That is why I have written in as 4c. And why is it deployed? Obviously till now no meiosis division has not taken place. It is actually called as what? Meiocyte, which will be going through the two successive process of the meiosis. Okay, now let me draw about the chromosomes. Let me show you what will be the exact point of view of the chromosomes, as compaction has taken place in the entire phases of the lactotin. Obviously, chromosome will be look more visible in this phase, isn't it? Let me show. Here I am using two color to know or to explain about the crossing over, okay, or the combinations. Clear? So centriole also start to move more towards the opposite pole, more towards. So in this phase, what will happen? In this phase, homologous pair with each other. You have seen if it is from the mother and this one supposed to be from the father, and these chromosomes are look like similar in shape, similar in positions of the centromere. I have made two different patterns of the chromosomes. You can correlate is metacentric, okay. Some metacentric also I can show you, but not necessarily. We need to know actually the exact procedures of the cell divisions. Okay. So what will happen so far? First, what will form? Synaptonymal complex will form. These things I have shown you. Now I am not going to tell you about the things. As synaptonymal complexes form, obviously synapses will be taken place. I am not going to define what is called as synapses, because already I have explained, shown also on the very beginning. So synapses. So you have seen the homologous chromosome pairs, which is called as synapses. It will be what? Which will form what? Bivalent has forms. If one chromosomes you correlate with the univalent, then two chromosomes will form what? Bivalent. And as four chromatids start to form, that is why it will form what? Tetrad. So chromosome tetrad you will see, bivalent also you have seen. Then what will happen? Homologous chromosomes pairs. So you have seen what are the things that's happening in the zygote. So can we enter into the next phase, which is called as what? Which is called as pachytene. Now packaging will be more pachytene will be taken place. Pachytene means what? Crossing over will be taken place. More packed, more packed. Now here you may notice or not, it is also called as tetrad. But tetrad is perfectly visible in the pachytene. But it is, if question will come, while in which phase tetrad has developed, you can write zygote. But perfectly seen in the what? Perfectly seen in the pachytene. Now let me draw the structures of the pachytene. The condition of the cell in the pachytene. Now another thing, what will be the ploidy of this? The ploidy of this cell obviously 2n and 4c. We'll keep tracking about the ploidy of the cell. And you have forgot or not, you have to actually start to dissociate or start to degenerate the nuclear membrane, because this one is going to ending. So the centriole migrated more towards the opposite pole, isn't it? Two centrioles, one will be what, perpendicular to each other, and see what will be the conditions that crossing over you will see. Let me draw homologous chromosome. Here what will happen first, a recombination nodule will form first. Suppose here recombination nodule, that recombination nodule will keep the that suppose there recombination nodule here, that recombination nodule will keep the point of the crossing over. Okay, here I will show you only one point of the crossing over. Here suppose recombination nodule has formed, then here will be the crossing over. Clear, I think. Here will be what, synaptonymal complex. So what will happen in this phase? Completely synapsed, because each one has fused with each others. Then four chromatids becomes perfectly visible. Recombination nodules also, I have shown you recombinations nodules. Crossing over occurs between non-sister chromatid. Non-sister chromatid, okay. Non-sister chromatid. See, let me show you in this figure. These things I have forgot to tell. See, non-sister chromatid. These are called as what? These are called as sister chromatid. What I told? Sister chromatid. Sister chromatid. This one also called as sister chromatid. So this chromatid and this chromatid are called as what? Non-sister chromatid. Non-sister chromatid. Very simple only. Okay. So non-sister chromatid will be from the homologous chromosome, not from the same chromosome. Same chromosome will be having sister chromatid, and non-sister chromatid will be another chromatids of the homologous pair or another chromosomes. Okay. So you have seen these are sister chromatid, these are also sister chromatid, but these two, if you will combine, these are called as non-sister chromatid. These things is going, it is actually telling. So what it is? So non-sister chromatid will be fused. So crossing over will be taken place. What? Crossing over will be taken place in between the two non-sister chromatid. So these things has happened, and here what will happen, genetic recombination is completed. Now another thing is, what is genetic recombinations? There you can I'm new term I'm going to use here, will be maybe reciprocal translocation also may taken place. So see, this is suppose that chromosomes after recombinations, you will see that here will be one part of that chromosomes will come, and here another part of the same chromosome has come here. There will be what this is called, this procedure is called as recombinations. Now in diplotin, what you are going to see in diplotin, let me draw the same cell in diplotin. Here I can show you that nuclear membrane start to disappear. That nuclear membrane start to disappear, and here nuclear membrane start to disappear. And what will happen as these are disappeared? Obviously, there will be, you will see one cross connection. That cross connection is called as chiasmata. Let me show you. This is the chromosome. This is the chromosomes. Here recombinations has taken place as synaptonymal complex has disappeared. That chiasmata will be seen perfectly clear. So I have not shown about the synaptonymal complex, and they have begin to separate except the crossing over site, begin to separate except the crossing over site, and exit point you have noticed this is called as chiasmata. And this diplotin stage actually it may last from month to years in case of vertebrates. In case of vertebrates, here human also included. That process of oogenesis in case of uh, fetus will be arrested in the diplotin stage, while that girl will be in the puberty. Then from the rest of the phases will be continue. That means what, in the diplotin stage it will be arrested, and while that girl, specific girls, will be entering into the puberty, from the diakinesis only oogenesis will start. That is why this is told about the, it is actually what, from 1 to 2 years in many vertebrates it may last. Now last phase of this Prophase 1, today in this video will be explaining only up to the diagenesis procedures, because later on what will be going to happen, it is very simple. Now see here, let me draw the nuclear membrane. Another thing, what will be the conditions of the centriole here? Obviously centriole also move towards more towards the pole. There will be the pole. The centriole start to move towards the opposite pole, and in this case centriole reach to the opposite pole, and they start to form spindle fiber. They start to form spindle fiber, because after the prophase, metaphase will be starting, and in this metaphase you will be seeing, in case of prophase, in case of meiosis 1, two metaphase plate you are going to see. Okay, so what will happen, see here terminalizations of the chiasmata will be taken place. Terminal of the chiasmata, if it will starting from this, if it will, this is the crossing point, and that terminalizations will be taken place. That means what? If it is the crossing point, then it will be ending and ending. This is the terminalizations point. Now what will happen? That chromosome, what will happen? What will be the conditions of this one? Here you have seen the terminalizations has taken place. Okay. Uh, Here you have seen, or you will see, the nuclear membrane totally disappearing. What will happen in the prophase of the mitosis? These things will be happening. All the organelles will be dissolved or disappear. Nuclear embryos break down and organelles disappear. Then homologous chromosomes not separated. They are not separated. In some many books it is written as separated. They have not separated. Slightly they have touched, and that separation will be taken place in the anaphase 1. Now let me show you what will be the condition. Okay, before going to enter, let me ask you the uh, ploidy nature of these cells. The ploidy nature of this leaf, it is 2n and 4c, isn't it? It is 2n and 4c. Here also, what will be the condition? It is also 2n and 4c. Now we have seen till now what? We have seen lactotin. In lactotin, what has happened? That chromosome compaction has taken place. Then chromosomes are visible in light microscope. And in zygotin, you have seen one synaptonymal complex has appeared. After that, two homologous chromosome fuse with each others, and that process is called as synapsis. And after that, bivalence tetrad, these things has happened. Then pachytene crossing over has taken place. Then first, what has happened? Recombination nodule has formed in the synaptonymal complex. Here another enzyme is held, which is called as recombinase. Okay, so that after crossing over, what will happen? That recombinations has taken place. Then in case of diplotin, synaptonymal complex start to disappear or dissolves, so that only chiasmata will be seen, and that chiasmata will be terminalized in diakinesis. So this is the entire story